Hello, good morning students. In continuation of our series of lecture on characterization of nanomaterials, today I am going to deliver a special lecture on an introduction to scanning electron microscope. It is in your course, M.Sc 4th semester student. Now, this is a very broad subject. I am planning to divide this lecture into two parts. In part 1, first I will discuss the principle of scanning electron microscope. I will not discuss the application of scanning electron microscope, or I will not discuss the difference between scanning electron microscope or transmission electron microscope. And in the second part, I am planning to discuss or demonstrate the working principle of scanning electron microscope. Fortunately, in this chemistry department, we are having a very sophisticated tabletop compact model of same, of course, Hitachi model. And I am really grateful to Professor Manas Kanti Dev, the coordinator of DST PERS project. You all know regarding the DST PERS, performance of the university research for scientific excellence. So the university was awarded rupees 10 crores of rupees, and large number of faculty members of our department, they are working on these various important aspects of the nanomaterials. And one of the prominent worker is Dr. Manmohan Lal Satnami. And I have taken some of the photographs from his research work, and I am also grateful to Professor Shams Parvez. So first, because you are all students, first I will explain the important parts of this scanning electron microscope, how it works, and then in second part, when you will see the demonstration of this scanning electron microscope, you will understand each and everything. And I will take the help of another very bright research scholar. His name is Rajiv Nayan. He is a student of another very good research worker of our department. His name is Dr. Indrapal Kharwal. And Dr. Kamlesh Srivas is also working on the material science. So it is a very important subject, and you all know that for any type of nanoparticle research, three things are very important. We must know the topography of that sample. We must know the shape of the sample, and we must know the crystal structure and other things. And in your course, we are having all such type of things. And I think in my first lecture, I have already discussed the scanning probe microscope, scanning tunneling microscope. I have already discussed the atomic force microscope. And you know that I am just using only few, but large number of techniques are available for the characterization. But today I will just only concentrate only on one technique, that is same, scanning electron microscope. And fortunately, sometimes our that present same model is integrated with this energy dispersed X-ray spectroscopy. So that means even elemental composition can also be determined with the help of that same model. And this is a very common information for the student, that how you characterize the nanomaterials. So please go through all these type of the system, that you do agree with me that there are large number of systems or instrument equipments are available for characterization of nanomaterials. As you know that this is an advancement, and I always consider that we are going from photons to the electrons, or the basic thing of the microscope, how optical microscope is converted into the electron microscope. And basically in optical microscope we use generally transparent lens, and in electron microscope generally we do use, I'll explain what is actually electromagnetic lens or electrostatic lenses. And of course I have already discussed, I will not discuss about the scanning probe microscope where sharp needle is required, that quantum tunneling current is required, and other things. Now, wherever I start or I teach any subjects, I always give very elementary information about the story of this invention. So although it is very difficult for me to discuss the whole story of the discovery of the electron microscope, but I must share this one of the most important research paper written by Macmullen. If you read this paper, you will have a information about the development of the scanning electron microscope, started from 1928 to 1965. And of course, during 1931 and 37, Ruska and Maxnall, they have actually started working on this scanning electron microscope. And in 1937, I have seen that one of the scientists, Manfred Erdiner, he actually invented a microscope with high resolution by scanning tunnel, a very small raster system. And of course, the first commercial scanning electron microscope was developed by, I think, this Cambridge Instrument Company in 1965. But if you think for the Nobel Prize on the subjects, so everybody is well aware of these things, that in 1986, 3 scientists were awarded Nobel Prize. These 2 scientists, you all know, I have already discussed this thing, Gerd Binning and Rohner, for this scanning tunneling microscope. But the 3 unitedly awarded Nobel Prize, and Ernest Ruska is the first electron microscope developed by the Ruska. And very recently, 2014, there is another Nobel Prize in the chemistry based on the development of super resolved fluorescence microscopy. I will not discuss all these things. So it is just for the student query, I am giving this information to the students. Now I am coming to the basic classifications, and in my previous lecture also I have shown this PPT before you, and I have already completed this. Now my today's concentration is only the scanning electron microscope. Today I will not discuss about the field emission scanning electron microscope, FESM. I will only concentrate on this scanning electron microscope. Now first, as you know that why you use this technique, and you will be happy to know that this is a very popular technique and very useful technique, and that has been used in various areas of the material science, in biology, electronics, as well as the nanotechnology. And due to its high resolution imaging and detailed surface morphology, it has been used most of the academic institution as well as some of the industries. And if you ask me, sir, what are the most important application of the same for any nanochemist wants to know about the topography, surface features of their sample or specimen, you want to know the size and shape, you want to know the compositions, amount of the elements, and lastly, of course, because from that same we'll get the three-dimensional impression. So, of course, some crystallographic information can also be obtained with this same. Now, let me explain in my second part, when I will show you the actual machine, you will understand the things. But there are three important principles. First principle is that here we will use an electron source. That means from some source, electric gun, electron beams are emerged. And we will also apply some accelerated voltage, and of course it depends upon machine to machine and as per your requirement. For our instrument where we are using, we usually use 20 kilovolt voltage. But in normal cases, it can be up to 30 kilovolt or 50 kilovolt. That is the first part. That means there must be an electron gun, and from there we are getting the electron beam, and through electromagnetic lenses and different apertures, this beam of electron is interacted with this sample. And after this interaction, you know that there is a emission of electrons. And third part is that when there is a different types of emission, I will explain that some emission or scattering is elastic scattering. There is no change in energy. There is some secondary scattering, secondary electrons or backscattered electrons like that. And that emitted electrons can be detected with some detectors. And of course, ultimately then with the help of this detector, we will figure out the image. That is the simple working principle of same, and sometimes it is TAME. I am not talking about the TAME here. Today I will not discuss the transmission electron microscope. In next lecture I will discuss about the transmission electron microscope. Now, whenever we are talking about any type of microscope, two part is very important. One is magnification, that means the distance between the two objects. That is also very important. And here magnification range is 0.3 million types of things. And of course, it is also depending upon the types of the instrument is also. And resolution is also important. I'm sorry, magnification means the enlargement of the image. And resolution means the smallest observable features of an image. And here is also, we can typically see between 0.5 to 4 nanometers, but because there is a compact desktop model where we are taking the 4 nanometers, and I think magnification is like 60,000, like that. Now the main type of analysis, what we will do. First thing is that we will detect, I will also explain the theory of that, what is actually secondary electrons. From the electron gun, we are getting the primary electrons. And when it interacts with that, different, you know that if you see structure of the atomic structure, there are different K, L, M, different types of shapes. There are some electrons. So we also monitor the secondary electrons, which is very important. Of course, sometimes we call it the low energy electrons. And this part is very important. Sometimes it's called the reflected or back scattered electrons, which is, of course, high energy electrons. And it is very useful to understand the in-depth studies. And fortunately, our instrument is very suitable for EDA study also, or sometimes we call it EDX, energy dispersive X-ray spectroscopy. So this is very important technique. You know that by that method we can understand the elemental compositions. And of course, we can also determine the cathodoluminescence or electron backscatter diffractions. Now I am coming to the main parts of the instruments. As I already discussed, that I am really grateful to our department and for providing me this instrument and giving me permission to work with this instrument. These are some research scholars. They are, as I told you, the large number of research scholars and faculty members, they are working on these things. And I will discuss. So you see, it is a very, very compact model. And of course, it is computer. This is the actual model. And I'll explain this. This is the sample place and where is the electron gone. There is a vacuum pump, everything is there. So in my next lecture, you will understand. And it is very easy to operate. That is the most important part. You see, separate research scholars, large number of research scholars. We don't have any operator for that. They are using this type of instruments. Now it is I think in undergraduate level also you have studied the electron microscope, and I think in second semester also you have studied these things. But here actually in the fourth semester, we are concerned about the application of scanning electron microscope. That is our main objective. But in order to understand the application, we must know some basic features of that thing. Now, first thing is that, of course, source of electron. So you should have an electron source. There is the electron gun, sometimes it is called. And our system is, of course, we are having the tungsten filament. But in some of the machine, they are having the LaB6, lanthanum hexaboride. And you know that another system is called the field emission gun. That is called the FESIM. We don't have that, but in IIT Bhilai they have FESIM. So this model, you know that it's based on this thermionic system. That is the first part. Now second part is the sample chamber. I will also explain what is sample chamber. And third is most important. If you compare with the simple microscope and the complex microscope. In simple microscope, there is a optical lens. But here there are two types of lens. One is condenser lens and second is the objective lens. Now, what is important that in this scanning electron microscope, both the lenses are electromagnetic lenses. That is the most important point. And in transmission electron microscopy also there. And in between this, I will explain what is the role of condenser lens, what is the role of objective lens. Basically, you can understand the convergence of the things. And in between condenser lens and objective lens, of course, there are certain aperture, because you know that whenever we are using different types of lens, they are having some problems, chromatic aberration, diffraction and other things. So in order to remove all those problems, there must be some aperture. And this is also very important in this scanning electron microscope. We use some scanning coils or sometimes it is called the deflector coils, which is also very important because sometimes it will be very, very useful to build the image. I will explain these things. And of course, as I told you that there may be some detectors, and in our instrument the detectors are different, and external vacuum pump, because the vacuum condition is very, very important. And of course, you see we are having two computers, two laptops, two computers. One is for the structure and another is for the exclusively for the EDX studies. And as I already discussed with you, that electron beam is emitted by the electron gun. So it is a very simple thing. Now I'm coming to main important point. Now sometimes what happened, you know that sometimes that your samples are non-conductive, and sometimes we use some biological samples, we do some studies on bacteria and other things. So in that particular case, we have to use one iron sputter coater. So when I will show you the instrument, you will see that there is a separate instrument that is called the sample preparation instrument to use with a scanning electron microscope, and it is also needed for conductive sample, and we will use very thin gold coating. And in particular our instrument, we are having gold system, but a thin metal coating such as platinum, sometimes gold, or platinum palladium alloy, or gold palladium alloy can also be used, and it is deposited on the sample. And then we'll put that sample, and in our system we can take 10 samples at a time, 10 samples at a time, 10 samples at a time. And so the surface of the sample becomes, sometimes what happens if you don't use that coating and other things, so there may be some charge and other things problems. So in order to avoid all those problems, we use the iron sputter coater, that is things. So I'll show you this instrument. Now regarding guns, I have already discussed with you that most of the time modern electron guns is field emission guns actually, or FESEM is definitely, FESEM is better than the SEM, but the cost is very high. Sometimes we use cold guns, a strong electric field is used. Sometimes the single crystal of tungsten, that also can also be used. Now my student Abhishek Katendra has prepared this. This is a schematic diagram, and it is available in the most of the literature available in the website. And as you see that start point is the electron gun. You can focus it because this is the most important thing. It is a electron gun, and this yellow color deliberately I have written it is a schematic diagram, and this blue color deliberately, this is, I'll show you that what is actually that electromagnetic lenses. Basically there are some copper coils like that, copper coils. If it is like copper coils, and there are some iron Fe poles of the iron, and this is objective lens, and then there must be some aperture, and this coil is very important, that is called the deflection coil. And there are different detector, black scattered electron detector, secondary electron detectors, EDX and everything. This is objective lens and the condenser lens, and this whole system is under the vacuum. Here particularly is this machine, I think it is Hitachi High-Tech Technologies. This is, you are seeing the vacuum pump, the sample chamber, and this is a special type of detector. Very good model, very handy. Any student can use this directly. Very small thing. The special detector is ultra variable pressure type of detector in this particular model. And as I told you that here we'll use 20, I'm sorry, it must be kilovolt. Typing mistake, it is kilovolt. But here we'll use the 20 kilovolt. But we can use, for normal system, we can use 5 to 10 kilovolt also. But for EDX studies, to use some high accelerated voltage. That means when there is an electron beam, it's accelerated by heavy voltage, and this is sample. And if it is transmission electron microscope, that there must be some, this electron transmitted through the sample, and we detect these things and other things, and everything is under the vacuum. Now this scan coil or sometimes deflected coils is very important. I'll tell you, because image is very important, and that scan coils, they use raster. It's a very important system. How we build that image, because there is a zigzag motion and other things. Now, how do this electron microscope work? I have already, I will not discuss any details things, that from the stream of electrons and accelerated them towards the sample. Of course, there is a cathode and anode, you know. Yes, there is the apertures, condensers, convert the image. Then, of course, detectors. And that coil, I told you that coil is there to raster-like pattern and collect the scattered electrons. Now I'll just very briefly, I'll not go into details of that, what is the basic mechanism of electromagnetic lens, because you know that there are some, as I told you, that these are some copper coils. And another side, in this side also, another, this type is there. Here is also that side. And there. So when we apply the voltage, so automatically it creates a magnetic field and other things. And there is some machine, they are having electrostatic lenses, but it's unusual. The way hand acts as a simple, but most of the cases, that is electromagnetic lenses. Now I must tell you that because we are in TEM sometimes condenser lens 1 and condenser lens 2, but here basically in our system, that condenser lens just below the electron gun, and adjust the diameter convergence of the electron beam, controls the current and intensity of the beam reaches to the specimen. Critical for optimizing resolution and signal to noise ratio. Sometimes that with the help of creating a vacuum, we can also control that signal to noise ratio. If there is a vacuum, there is no other gases, no question of scattering and other things. Whereas the objective lens focuses the electron beam precisely onto the specimen. plays a major role in achieving fine beam spot size for high resolution imaging. So this is the role of condenser lens, and again in some it is lenses, LEN, lenses. So spelling mistake may be there. Lenses. Now the role of it is very important. Just bear in mind that near this objective lens there is a scanning coil or deflection coil, and the role of this deflection coil deflects the focused beam in horizontal and vertical directions, covering the surface, the sample in raster pattern. If you want to learn much about the raster, what is actually raster pattern, you have to read some of the scientific paper. You will get complete information about what is the raster pattern, or in one sentence you can write that is essential for image formation, because at each scan point correspond to one pixel in the final image, because there may be some zigzag things. So to create an image of a small surface of a sample by firing a high intensity beam of electrons at the sample surface and scanning in a zigzag type of the pattern, or you can call it the deflection system raster the electron beam over the sample and allow an image to be formed. Because here we are doing nothing. Electron beams are coming from electron gun through the objective lens and condenser lens and interact with that sample and emission of electrons, scattering of the electrons, and we detect those electrons. But we are interested in the shape and size of that particular nanomaterial, so therefore this raster pattern can be obtained with the help of this deflection coil. That is the role of the deflection coil. Now I think this is the last part. We have already seen all these things that I am already told you everything. Now I will confine that very briefly what is secondary electrons, what is backscattered electrons, and what is X-ray and other things, because everything is clear now that high energy electron beam is there is interaction and production of, there is a very good, again it is a block diagram, or you can call it a schematic diagram, and in different types of books it is like this is a sample and this is primary. If I talk, it is a primary electron beam, and then there may be different types of things, agar electrons and other things, and you have to monitor all these type of, whether it is a secondary electrons or backscattered electrons or whatever it is. So we have to monitor all these things and get the final composition, structure and other things. So again my students has prepared this very beautiful diagram, and you can call it a sample volume element, and this is electron beam, or sometimes it is called the primary electron. This is the primary electron, and it interacted with these things. I'll explain what is secondary, what is backscattered, but bear in mind that secondary electrons are low energy electrons and inelastic scattering. That means that primary electron donates the energy to them, but whereas backscattered electrons are results of the elastic collision. There is no exchange of the energy, and it is high energy things. And second point is that basically if you detect the secondary electrons, it can monitor the superficial part, surface area, and depth of this backside electron high energy. So therefore you will get some morphology. And of course, most important thing is that energy disperse X-ray spectroscopy, and that will give the elemental compositions. And when we perform the experiment today evening with our instrument, with some nickel sample, you will see the actual composition of nickel. There is a gold plating, so you will also see the gold composition and other things. So this is the most common. I'm not interested in this inelastic scattering composition, incoherent elastic scattering. There are other things, but there are two detectors basically, and sometimes we will also monitor the mixed secondary electrons as well as the backscattered electrons and other things. So that can also be explained by that way. It's a very simple. Again, it is a block diagram prepared by a student. That it is a the simple atomic structure, and after this interaction with the primary electron with these secondary electrons, so naturally secondary electrons is emitted. We have to detect these things. So the path is changing. So therefore I have written a new trajectory. But unfortunately this is inelastic scattering. And I think only the electrons, low energy electrons, I think 50 electron volt, less than 50 electron volt. And here 180 changes. And that is called the backscattered electrons. It's a reflection of other things. So you see, it is elastic scattering. I have told you many times, no energy change. Here is the energy change. And here also a new trajectory, and that can be monitored and get the information about the topography, morphology, composition and other things. So just for the student, I have written these things that secondary electrons are low energy electrons and less than 50 electron volt, and originating from the outer cells of atoms of the sample. These electrons are emitted due to inelastic scattering, and because that primary electron transfers part of their energy to the sample, and that can be ejected from the sample of the surface, and that is created by the detector. And these detectors can only travel very short distance within the material because only surface. As a result, the detectable secondary electron signals come from a very shallow region near the surface, typically within the top 10 nanometers. The shallow escape depth makes secondary electron imaging exceptionally sensitive to surface topography and fine structural features, providing high resolution image of surface morphology. At the end of this lecture, I will show only two pictures of our own system. One is magnesium oxide, manganese oxide, and I think cerium oxide like this. You will see how beautiful structure can be obtained with the help of SEM. One is flower type of the structure. Now second is the back scattered electrons. This is of course high energy electrons and elastic scattering. One of the most important thing is that these electrons originate from deeper regions of the sample compared to the secondary electrons. I am not saying that there may be some, when you go into details of the literature, good books, you will find AC1, AC2, secondary electron 1, secondary electron 2, secondary electron 3, and like that, and mixed of AC and BAC. What is important is that, this is very important, the student must note it, that for the backscattered or reflected imaging, it depends upon the atomic number. Because we are using different types of element. So heavier element, those with a higher atomic number scatter electrons are more effectively, resulting in brighter regions in backscattered electron image. Whereas lighter elements, lower atomic number, scatter electron less effectively, producing darker areas. So these makes backscattered electrons imaging especially useful for distinguishing between different phases in a sample, compositional mapping, and detecting inclusion of foreign particles in a matrix. Therefore, in industry, this is a very useful technique. Of course, nowadays people are using FESM, transmission electron microscope, and other things. Now, the last part is that we detect the secondary electron, we detect the backscattered electrons. Now, we also detect that energy dispersive X-ray. So, EDS or EDX is an essential analytical technique. You know that there are two types of energy dispersed X-ray, wavelength dispersed X-ray, because it will provide the most important information about the elemental compositions. And of course, it will identify the elements present in specific regions of the sample. So, it is visible, green color. When the light energy electron beam in the same interacts with the atom of the sample, it can knock inner cell electrons out of their typically K cell or L cells. This creates an unstable state and the atom relaxes by having an electron from a higher energy cell. Each element emits X-ray at unique energies that correspond to its atomic structure, allowing for identifications. And when I perform the experiment with the nickel sample coated with the gold, as it is already mentioned that each element, gold different, nickel is different, so you will get two very important information like that. So now everything is okay, our experiment is completed. Now the formation of the image, and you know that we have to detect the signals, and we are having very good computer. So the image in a scanning electron microscope is obtained by scanning a focused beam of high energy electrons across the surface of sample and detecting the signals. And then, as I know that the raster process scanning these things, detectors, secondary electrons, backscattered electrons. Of course, the name of that detector is Everhart Thornley detector, named after some two scientists actually developed this thing. So I will not go into details of that, the model of the detectors. But detectors collect the emitted electrons, and then the intensity of the detected signal varies depending on the surface features, material composition, and the topography. And you will get the final image of these things. So the detected signal from each scan point is converted into brightness value for the corresponding pixel. When in second lecture, with the help of Mr. Rajiv Nan, we perform this experiment, now you will see that how that picture or figure can be obtained with the help of computer. Everything is now is modernized and mechanized. Everything with your hand. You can monitor these things. The pixel values are compiled in synchronizing with the beam position to create a grayscale image on the computer monitor. You can get different colors or whatever it is, brightness and other things, size and everything, size and shape, topography, everything. So brighter regions indicate higher electron yield, raised features of heavy elements, while darker regions, as I have already told you. So this is all about the very, very elementary information about the scanning electron microscope. My dear young friends, I am not giving you the information about the difference between the scanning electron microscope and transmission electron. Also, this is very important. You must know what are the features that are similar to the same and same, and what are the different features. And I will not go into details about the other application part or the demerits, nothing. And there are many examples available in our literature, but here, because it is very difficult for me to show all the pictures. Basically, my student now, he is a very vibrant faculty member of our department. His name is Dr. Manmohanlal Satnami, and he has actually studied large number of nanomaterials, starting from metallic nanoparticle as well as the semiconductor nanoparticle, cadmium sulfide and other things. So I requested, he did his PhD under my guidance. I requested one of our students, Yogyata Chaure, that for the student, you must give the two best, your 10 pictures. The morning, she has provided me 2 pictures. One is, you can see the same image of manganese oxide nanowires. You see how beautiful it is shown. And one important thing is that it is published one of the most important journal, ACS, Applied Nanomaterials and other things. And second, you see the beautiful flower type of things, cerium oxide nano flower, how clear it is. So it is just 1 or 2 examples. There are a large number of examples are available in literature, in the website. Students, I think many students of you are now working M.Sc research projects, and I am very happy that large number of students are working on the nanomaterials. So I will urge all the students that you prepare simply, of course, gold and silver and nanorods, different types of gold nanorods are also available in the literature. But for clarity and novelty, I have shown these 2 pictures of Dr. Manmohanlal Satnami's work. I hope that with this first part, you have got some elementary idea about the different parts of the scanning electron microscope. What is the role of electromagnetic lenses? What is the role of deflection coil? What is the role of detector? Now, after this elementary knowledge, when you perform the experiment in next part, because this is the first part, in second part, I'll go to the upstairs. There we are having this Hitachi scanning electron microscope with the help of Mr. Rajiv Nayan. I'll do some experiment and show you the actual working of the machine. Thank you very much.